I'm doing very well. Early morning here on the East Coast of the US, but all is well.
Where is it on the East Coast in the US?
Outside of Philadelphia, we're a
Oh, beautiful.
Philadelphia-based company. I was an academic faculty member at the University of Pennsylvania for many years after training there. And we started the company just sort of down the street from the university where we could take advantage of all the resources and core facilities and collaborators that we'd cultivated for so many years.
Karin, I read in your material you spent over 2 decades in cancer research. That's amazing.
Yes, I'm a cancer nerd from top to bottom. I eat, sleep, breathe, live it 100%.
That's fantastic. Let's jump right into the questions. So you spent 2 decades in academia and then decided to jump ship and become an entrepreneur. What motivated you to have to make this decision?
I think there's two components to it really. One was my just the overwhelming feeling, to use the metaphor of the war on cancer, that I felt like a soldier that had trained and trained and trained for a battle and never got to fight it. And that's frustrating. You train as a cancer biologist to learn everything you possibly can, and you want to get out there and fight the fight. But it's not a fight that you can deploy in from an academic chair. That really has become a very it's about the training. It's about training the next generation. It's about learning and studying. And to get out there and really fight the fight meant starting the company and to fight the fight in the way that I wanted to. And so that really was the motivation. And then, of course, there's the timing and the consideration of when did I find my thing? And I had this conversation with some great academic faculty that I thought should have started companies long ago. And I said, why did you never jump in the ring? Why did you never start a company? And the response was, I never found my thing. I never found the thing that I could really hang my hat on and say, this is me. This is the idea I want to bring to the market. This is what I think is going to have the most impact. And I think I'm one of the fortunate ones that did find my thing. And that really coincided with that growing frustration that, I'm constantly preparing for a battle I'm never going to get to fight if I don't make change.
You see research as constantly fighting a battle, but you're not really participating in the battle.
Not anymore. I think there was a time, I came up in an era where we all thought we're going to publish our best ideas and the pharmaceutical companies are going to come along and they're going to license it from us and they're going to take it to the next step. And that, our contribution is the publication. Our contribution is getting the idea out there. And I
Mm.
think that was perhaps true at one time, maybe not. Maybe it's just what they told us as part of our indoctrination as graduate students. But I think it's certainly not the case now. Now there's, the literature, the growth in human knowledge is exponential. As I often tell people, we can't sit around waiting for the Pfizer fairy or the Novartis fairy to come around and, hand pick our technology and just, instinctively know that that's what's going to be critical to change the field. We have to get out there and do it ourselves. And now that I've done it, I see very much that the people who are potential investors, what they care about is that the founder believes so strongly in what they're doing that they were willing to get off out of the academic chair and move into the space to pursue it. They're looking for that passion and that commitment as a vetting of the idea. And so perhaps there never was a Pfizer fairy that was going to come along and commercialize our idea, but there certainly is not one now. And when you find your thing, that's when you really have to commit. And that's what I did.
Yeah, that's true. You mentioned that you found your thing. What's your thing now?
The thing is our platform at SyzOnc. So we founded SyzOnc in 2023, raised some pre-seed capital, and really what we're doing is we created a platform and a therapeutic strategy that is inclusive in that it really reflects the true state of solid tumor biology and leaves behind a lot of the dogma and fallacies and misconceptions from the past, and along those lines, we develop technology and tools that would allow us to reveal truly the most important therapeutic targets and the
Mm.
best ways to pursue them.
That's good to hear. I mean, when I think about the patient side, cancer is usually when they get a diagnosis, it's a moment of fear and desperation, and it's not a nice thing. How do you deal with that in your work?
Yeah. For me, that's the critical piece of it. When I think about what I'm really fighting, the battle that I really trained to fight, it is that the fight against that fear, that helplessness, that powerlessness, that something you get that diagnosis and you panic and everybody around you panics, and everyone is afraid and it ruins lives and it takes lives. And that really fundamentally is the fight that I'm fighting. The name of that fight happens to be cancer, but it's that curse on our society that takes away joy, that takes away parents from children and children from parents. That's the fight we're fighting, and that's why I don't struggle to go to work every day. That's why I don't lose hope. That's why the bad days or the tough days don't they don't get me down. Because when you're fighting that fight, you keep going no matter what because the fight is worth it. The mission is true and valuable.
Yeah, that's true. That's true. I know some people who suffer from the disease, and it's really devastating when you see how it progresses.
Yeah. Yeah, I agree. To me, it's inspiration. It's what keeps me going. Our patient communities that we interact with, that look to us, knowing that we probably can't save their lives, but we can save the next life if we work hard and we work together. Those are those inspirational moments that I always reflect on a tough day. That, you know, this person lost their life in the fight, but all they wanted to do was come see the lab. All they wanted to do was see our progress and talk about what the future held. And if they can do it, then I definitely can.
So, what changed basically between academia and now as an entrepreneur is that you have more exposure to patients, to real patients.
I don't think more. I just think that because as an academic, we were constantly touring the lab, we were constantly interacting with our patient communities. And I think that's part of the beauty of academia is that you do have patients that are part of your community as part of the hospital systems, and they fundraise and they're part of your fight. I think what I feel now is that I'm a better effector for their mission, than on the front lines of that fight. And so I feel like I'm carrying their message forward more effectively, and we're getting closer and closer to changing the present and the future in their honor.
If you compare your life in academia and now as a CEO of a company, what did change for you the most?
Yeah, it's really interesting. Fundamentally, it's the same job. You're doing science, talking about science, and raising money to do science. Those are the three main components. But as a CEO, well, let me say, as an academic, you have to be in the present and you have to kind of have 10% of you focused on what's next and the future, because you really have to be present to mentor students, to get your papers published, to write today's grant. As a CEO, you have to be 50% in the future, 3 months ahead, 6 months ahead, a year ahead, because I'm out there in front of everything that we're doing, making sure that all the relationships we need are in place, that the tools and CROs and techniques that we're going to need 6 months from now, I can't start prepping for that 6 months from now. I have to do it now. So I'm much more future focused as I'm juggling all the balls that are associated with leading a company than I ever was as an academic. It changes like literally you go from a present tense person to a future tense person overnight. It's really stunning. And I think either you can adapt to that or you can't. And that might be the fundamental go, no go, fail, succeed, defining point is whether you can make that switch, because it's really challenging to put yourself in the future and say, what are we going to need then? Because I got to start working on it now. And you just don't do that as an academic. You are very much dealing with today's problems.
Before we dive into cancer and what cancer is and how we can cure it, let's stay a little bit on the transition from academia into entrepreneurship, because I know that many people think about it but
Yeah.
have no clue what that means. And there was one point that you mentioned that the switch from academia to business life meant going from being a person in the now and present, which is a very Zen-like what's the attitude, to becoming a future person. And this is the first time that I hear that. What do you associate with being a future person?
It's almost like having a crystal ball, right? You have to, just a hard and real example, we're developing a drug. What will I need? Where will I be in 3 months to take that drug to the next level? And so I need to, move those pieces into place now, starting that, so that they'll be there. So, for example, if I need um, generative AI, if I'm going to need generative chemistry in 3 months for a molecule's development, I need to start vetting those companies and interviewing those companies right now. I need to get the paperwork done. I need to have a services agreement in place. When my CSO comes to me and says, I'm ready for generative AI, I'm ready to do the generative chemistry, I can't be at that point just starting to talk to those companies and trying to figure out who the right person to talk to, because I'll delay her by 3 months every time I make a mistake like that. And I can't afford those mistakes. We can't afford to lose that time. We can't afford to the financial cost of it. So I have to be in her shoes in 3 months and and 6 months and a year from now, so that I'm providing, as she tries to take the next step, I'm putting the stone in front of her so she can jump to it, right? That's my job, is to create the path to to drug development. And if I'm not thinking about that every day, what's the next step? Who do I need to talk to? Which services do I need to get in place? She's going to falter. She's going to find roadblocks. She's going to have delays. And that's what we have to avoid. And that's a completely new thought process for me. It's just not something that comes up in the course of academic life.
Why doesn't it come up in the course of an academic life?
I I guess because so many of the services that you'll need as an academic are just they're right in front of you. That's what the university is. It's a community of resources. And so if you think in 6 weeks you're going to need a single cell sequencing core, you email the core and say, "Hey, I'm going to have a sample for you in a couple weeks. What do I need to do?" And they say, "Drop it off," and that's it. It's not a There's no master services agreement. There are no lawyers. There's no complexity to it. It's just, show up, maybe we have one meeting in advance just so you can tell us what your science is. It's just a very fluid environment. And that's the beauty of that environment, is that everything is all around you and it's all provided as part of the community. But when you're out there on your own, as an entrepreneur and building a company, the services that you need they're not in the building with you. They're in China or in India. And there's many of them to choose from, and you need advice and you need guidance and you need to speak to references and you need to talk to your lawyer about it and you need to get contracts going. All of that complexity is just not part of academic life. And that really is the silver lining of it. That's the beauty of it. That's what it's for. But at the same time, they're not using CROs to synthesize molecules, right? If you want a molecule synthesized in academia, you need to start a collaboration with a medicinal chemist in the Department of Chemistry. And of course, the difference there is you're going to wait maybe 2 years to get your molecule because of, they have a lot of projects and they have their own problems. Whereas in the business world, everything moves incredibly fast. You sign a contract with a CRO, they immediately begin to work on your project, and 6 weeks later you have your molecules and you move forward. So the pace of things is very different, and you pay for that by having to be a futurist, by having to live in this perpetual, how do I set up these parameters, how do I create this infrastructure? And creating infrastructure is just not something that academics do on the regular.
Yeah, it's definitely the right approach to think about the future, what the patients need in the future, and then plan backwards to now and provide the resources to the team and get the team in place that your science can move forward.
As quickly as possible. and as well as possible without, making mistakes. And, this CRO didn't work out and it cost me 6 months and I have to start with a new one and those kinds of things.
Elon Musk and Jensen Huang are well known for urgency in their businesses, electric vehicles and chips, to put it bluntly. Do you think we can we can create this sense of urgency and speed in drug development?
I think we are creating it. I think I don't know if it's just the pace of things in general in life has become so fast that we want to catch up to it, or if it's the tools, the expansion of machine learning and AI and tools that make it possible. But we are in that urgency phase where um, you know, the key is accelerating certain processes, getting away from spending $10 million in 2 years to identify a hit, doing that faster, doing it better. Um, I think we are in that age of urgency for therapeutics. And maybe it has to do with, financial incentives and competition on all sides, and, the way that international geopolitics and economics have sort of condensed around us to create that urgency. I'm sure all of it matters and all of it has had an impact, but I think we are living in that age of urgency around therapeutics.
Yeah, no, we definitely need that. And let's move to talking about cancer. It's good that you are on the show. I would like to understand the disease better and what it
Yeah.
is and why it's so hard to cure it. There was this interview a few years ago, I think. It was, I hope I get it right, the CEO of Alphabet who said in 10 years, generative AI will develop a molecule that cures cancer all over the world. Do you really think that's possible?
I think it's possible that AI generative chemistry will produce that molecule that's really effective. But I'm a strong believer in combination therapies, and I think that the idea that one tool, there'd be one magic pill that people all over can take, that cures cancer, that's not a realistic goal. But I do think that AI is accelerating the drug discovery and drug development process to the point where I think we will get there, where it'll be a mostly AI generated molecule, that is foundational to a lot of treatment strategies.
Mm. No, it's definitely helpful, generative AI, but when I look currently on the market, I think there is a lot of sales talk out there. What will change?
Yeah.
Exaggerated timelines. It feels like going back to the 90s with the internet. And I have a hard time imagining that one molecule is this, let's just say, golden pill that will, or golden goose that will solve
Um,
all problems. What is cancer? What is this disease? We use the term broadly.
yeah, we use the term broadly, and maybe we shouldn't, but because it's many, many diseases. Each tissue type is a different cancer. And within that tissue type, there are usually multiple, if not many, different kinds of cancer. So, for example, my background is in soft tissue sarcomas. At this point, conservatively, people estimate that there are 100 different subtypes of soft tissue sarcoma, each one with its unique features, each one coming from a unique tissue type. And to lump them all together is not effective. Knowing where they can be grouped together is an important part of clinical trial development. But that's really the complexity and heterogeneity of cancer, is that it's not one thing. And I do think that heterogeneity is a massive part of why we struggle, why it's so difficult to cure. And I always tell people, right now people just say cancer. It's my goal that in 5 years, people will say liquid cancer or solid cancer, or liquid tumor versus solid tumor. Those two distinctions are massive, and people don't even say that, So it's there's a balance that we need between generalizing so everyone understands and overgeneralizing so that the complexity is lost. And I think our human aversion to complexity is also part of the huge problem in cancer biology. We want things to be simple by nature. We want things to be able to be reduced to simple experimental models to simple conclusions. And cancer defeats that goal. It completely overwhelms that human need to keep things simple with its complexity. And until we accept that complexity and dive into it and lean into it, I think we'll continue to struggle.
You see it as a human need, simplicity.
100%. Um, and there's so many examples of where cancer biologists in their daily experimental approach, they know something to be true. They know that there's an added layer of complexity to something, um, and they won't incorporate it into their systems because we believe in reductionist models. We believe in that in the simplest Occam's razor, right? The simplest answer, all things being equal, is generally the right one. And cancer defies that. It defies Occam's razor. It defies our need for reductionist tools and simple answers. It simply won't cooperate with that theory, with that approach to life. And as you know, we can't make it fit into that box no matter what we do. And I think that sums up what we've been doing for the last 30 years is trying to force cancer into that box, into something that cooperates with us. And that really is the source of our failures.
Mm. What's besides simplicity and complexity? I mean, when you look at drug development in the last 30 years, there was a lot of development, and when I look at other disease areas, we found cure. But cancer as a whole, when I look in the last 20 years, is still the same challenge. There is
Yeah.
some slight improvements, but I can't remember if there was a huge breakthrough where we can say, okay, now we found sort of golden pill. What's the biggest bottleneck in your opinion in evolving cancer research and then later also the clinical part drug development? Where do we struggle currently?
Yeah. I think that there are misconceptions in the field, even among experts, that have created about an ideological bottleneck. And when I say that, I mean we have all, since the human genome was solved, the last 20 years, we have all been tunnel vision, looking at tumors as genetic diseases. There must be a mutation. If we can't find a mutation, we need to sequence deeper. Just unwilling to change our ideology quick enough to integrate everything that all the information that's coming at us so rapidly. And it's created this backlash of people doubling down on old targets, old ideas, because they can't handle the uh, the complexity. And so for example, in many solid tumors, there are no oncogenic mutations that drive the tumors. They're completely driven by environmental processes, the lack of oxygen. I mean, they may start from a genetic change, like a loss of a tumor suppressor, but ultimately, the growth of the tumor and the spread of the tumor is governed by environmental properties, not external environmental properties, but in the properties of the tumor. And those are things that people simply don't know how to include in their models. And even if they do know how to include it, it's too complex or it's too expensive. And so this commitment, this to keep our heads in the sand instead of looking to change and looking to grow has been a real problem for target discovery. Drug discovery is a different thing. I think there is much more willingness to adopt the new tools. Anything that accelerates drug discovery catches people's interest. And so I think that's where AI and ML come in, where, the predictive tools for looking at a small molecule and being able to very quickly assess its toxicity properties and its permeability properties and all those things, those are tools that drug discoverers and drug hunters are very happy to engage with. They are very happy to make a process less expensive and faster. It's target discovery that has fallen so far behind, and that's why, as you say, nothing has really changed. Because people have stuck to their old interpretations and look at the same targets over and over again, unwilling to broaden their view of tumors to what we focus on, which is the tumor as an ecosystem, to stop looking at individual cancer cells, individual mutations, and just the cancer cells alone or just the immune cells alone. But this is a highly integrated network of features and environmental factors that if we don't take a system, we have to take a systems level approach to it. And we haven't been, and that's really been the bottleneck.
Wow, there's a lot to unpack in what you said. There's a lot of points where I would like to dive deeper.
Mhm.
Uh, let's start with the first one. You mentioned earlier that you see the bottleneck in cancer research as an ideological problem.
I do, yeah.
I always thought scientists are open, they are used to debate, they really destroy what doesn't work and are not really easy to trap into ideology. Why did it happen that we just focus on genetic mutations?
Such a good question. The human genome was solved. It was a massive achievement, still is a massive achievement. It taught us so much. It gave us new tools. And I mean, to say that we spent 20 years on it, I'm not sure that any revolution or innovation like that would have been treated any different. Now, looking back on it, it seems like too much. It seems like we have to move on to the next thing. But it's a typical human response, right? We have this massive change in our understanding of fundamental biology, and we spent 20 years deep diving into it, thinking that it was going to solve all our problems. And I think the immunorevolution was very similar in terms of the time that maybe a shorter amount of time has gone into it, but it was the same inclination. The immunorevolution changed the way everybody thought about therapeutics and tumor biology, cell therapy, changed the world and everyone's perspectives. And I think maybe we've been able to prove and disprove where it works and doesn't work much faster. And so it doesn't seem like we've wasted any time. It seems like we learned really quickly what works and what doesn't work. I don't know if Carl June feels like it was quick or not. That's up to him. But for some reason, the solving of the Human Genome Project, that I guess because it was so massive, you're talking about 30,000 genes, and who knows, that it takes a long time to rule those things in or out. Um, and it overtook everything for so long. And then when an entire generation of cancer biologists comes up under that ideology, it takes a long time to undo that kind of, I won't call it an obsession, but that sort of one-dimensional thinking. Before that, before the Human Genome Project was solved, perhaps people were more likely to pursue lots of different routes of discovery, lots of different areas of inquiry. And then the Human Genome Project was solved, and it became everything all at once, all the time. And I think a generation was changed because of it, for better or for worse.
So basically, you say that people living under this ideology, looking classically on finding an old target, an existing target, looking at genes, finding something that matches in the lab, that they can treat in the lab, cell line, and then try to make a drug out of it.
Yeah.
And you earlier mentioned that this is a wrong approach and you want to change it on a system level, and that your company is looking more on, you mentioned the term, it's a new systems level approach to drug discovery. What does that mean?
Yeah. So when I say system, when anybody says system, they mean a complex network, right? It's not just one thing. We're not just looking at the cancer cells themselves or the mutation or a given mutation. We're looking at the way that individual components of the network interact with one another. So when we're trying to identify the best target, we look at all the data available, and it's multimodal data. We don't rely on a single data type. That I think also is not the right approach. So we look at mRNA level data, we look at protein level data, we look at immune level data, we look at tumor infrastructure data, and we say, what are the targets that really represent critical nodes in those interactions that are governing multiple aspects of the tumor itself? those are the targets we want to go after. I don't want to go after simplistic proliferation regulators, cancer cell proliferation regulators, because I know that the tumor is an evolving dynamic system. And as soon as I hit one of those targets, it's downregulating that approach, that proliferation pathway, and upregulating a new one. It doesn't care that I want to kill it. It's only looking to survive. It's a basic survival approach, right? So we need targets that are so powerful and sit at these critical hubs in the ecosystem, in the network of the tumor, that the tumor cannot evolve very quickly a pathway to overcome. That's the difference. In those old targets that started with, MAP kinase and EGFR and RAS, and to this day, people are, there's still an obsession with mutant RAS inhibitors, and they have their place for, for certain, not the least of which is they're incredibly educational tools, and hopefully help some patients. But in reality, that's a very, very basic cancer proliferation inhibitor that would took decades to innovate and finally create. And everyone is so excited about it. But at the end of the day, you're simply regulating a very basic process that the tumor can evolve past in 3 months. And that's ultimately what happens with many of these inhibitors is you get some amount of, you've knocked the tumor back for 3 months or 6 months or a year, and then it comes roaring back in a way that you can't necessarily predict very well, because there's so many options. And then you have a worse situation. You have a resistant tumor, and a patient who's essentially going to have a poor outcome because of it. Those targets of the past, um, have we know what they're capable of, we know what their limits are now, and the future is about, we have to move past that and look for these targets that are much more critical power players in the tumor that the tumor cannot evolve beyond.
Does that mean that you criticize in the entire industry that we don't understand biology well enough?
No, I think we do understand it. It's that human reluctance to incorporate everything we know. I
Mm,
think so much powerful research has gone into the various levels of the system, from extracellular matrix architecture, immune evasion, T cell function, macrophage function, cancer cell features, oxygen responses, nutrient responses. We do know so much of what we need to know. We simply lack the wherewithal to deploy it. And that's what we're trying to do is not protect ourselves from the complexity and say we welcome this complexity. This complexity is the problem, but by acknowledging it is the only way we're going to overcome it. And looking at the tumor as an ecosystem, and actually deploying all the information that we do have. And I think the best example of our reluctance to do this, comes from the field of hypoxia or low oxygen signaling. And I mean, everybody knows about in the tumor world, everyone knows that tumors are hypoxic, that they outgrow their blood supply. And so there's areas of the tumor that are very, very oxygen deprived, and that has a significant effect on tumor cells. And this is so well recognized that the field of hypoxia was awarded the Nobel Prize several years ago.
mm, mm.
Three independent investigators who had solved various problems and now understand very well, how cells sense oxygen. And they're all cancer biologists. They all study this in the context of solid tumors. But if you ask any solid tumor biologist whether their experimental approach includes monitoring oxygen levels or testing under various oxygen conditions, including hypoxia, 99.9% will say absolutely not.
Right.
Absolutely not. They do all their experimentation at normal oxygen concentrations, knowing full well to the point of a Nobel Prize that oxygen concentrations are critical, they're low in tumors, they impact the tumor cell, they impact everything. Why don't we incorporate this knowledge into our systems, into our tools? We don't do it. And that's what I say, it's a human reluctance to embrace the complexity. It's not that we don't know it's true, right?
Mm,
But absolutely has not, something so basic hasn't been incorporated into our modeling. And I think, and this to me is the major problem. And it's perfectly natural for us to avoid that complexity, right? It's our brains as scientists are telling us that we need to pull apart a system and test each part of the system to understand it thoroughly. And then, but what we forget to do is put it all back together at the end, right? And create a three-dimensional picture of the situation. That fundamentally is where we fail, not as scientists, as humans.
That's an interesting thought. So your point is when we look at the car, for example, to make it understandable for people who are not in science, that scientists love taking cars apart, looking at the single parts in the system and then just leave it there and just lay it out on the table, and that's it. What they don't do is finding ways putting the car back together in a better way in the end of the day. And this is the situation in cancer research. So to understand the part, there's a lot of data out there,
Mhm.
but putting this data then to work to develop better drugs, find better targets, it's not happening at the scale it could happen.
Right. That's what I see
Mm,
as being fundamentally what's going on. And the reason why it's now is the time to talk about it is because we are at this critical tipping point where we know so much and science has produced so much technology and results and information and characterized tumors, especially certain tumors like breast cancer, for example, so well that we really are in a position where if we put all the information together and create better models and look for those targets, we will find them. We are at this precipice where we can completely change the game because of all the incredible work that has gone into the last 3 decades. I mean, it's just been a powerhouse. The explosion in findings and literature, the scientific literature is massive. And so all this hard work has gone into it, and where we're failing as at the point of making sense of it and deploying it and integrating. And that's the hardest part.
mm,
I would think about, if we try to build a rocket ship to go to Mars and all we care about is the rocket fuel, you're going to really successfully launch, but you're not
mm,
going to get there, right?
mm,
Because you're going to have ignored all the other components. And that's kind of where we are as cancer biologists, is we put so much time and energy into our rocket fuel, into our proliferation mechanisms and understanding what drives a cancer cell or what drives a T cell, and haven't there's been just we don't have the integration. We don't have the three-dimensional pictures
mm.
of each individual tumor that will give us the targets that we need. But we have the information, we have the building blocks for the first time in the history of humankind. We have the building blocks to put together the whole picture, to see the puzzle, to see the picture we're trying to create. And all we lack is the will and the know-how to do it, to build those. And I think that's why AI is having a moment right now, because people are thinking about using all the data we have to create that integration. We're not there yet. We struggle to integrate, we struggle to put the puzzle together, but the pieces are there. That's what's so amazing about this particular moment.
Yeah, great visions. I mean, it comes Elon Musk and says, let's not look at rocket fuel, but save humanity and several levels, and then things start to change. When I go back to your amazing story, you grew up in the system and basically were trained like any other person, any other scientist. And then at one point you came to the realization that's not going to work. There is something wrong and I want to change it. How was that moment for you? Is there a special revelation, a special moment, a special conversation that you remember?
Um, it's definitely the accumulation of a lot of moments. One thing that sticks out in my head is submitting an internal proposal to my institution, where you'd think, okay, this is a high reward, high risk, high reward situation. I'm not going to the NCI for incremental funding, where they look at things as like, are you going to get a paper out of this? Is this going to work? If we're 99% sure this is going to work, we'll give you the money. It was an institutional award where I was looking for money for a pilot of a new idea, high risk, high reward. And the response from one of the reviewers is, we shouldn't give her this money. This is never going to get an NIH award. Too novel, too avant-garde, too unprecedented, thinking about the ecosystem, thinking about a system level approach. And for me, that was a formative moment because I thought, isn't the institution supposed to be about innovation? Isn't that what we're doing here? And what I realized is everyone is trying to survive in a really, what even then was a really tough funding environment. And that environment, research is a living thing. It's dynamic and it responds to external pressures. And when the external pressure is toward incremental improvements and publishable factoids that have just been highly tested and are rigorous, but are nevertheless incremental, uh, that pressure changes the way we do science, changes the way we think, and it creates a an abyss where even the most adventurous scientists feel like they have to stick in a very well-established paradigm and play the game in order to stay funded. And maybe when they're 25 years into their career and full professors, they'll have enough, strength or support where they can try something new. And to me, that is it's the road to nowhere. We need to invert that script, in my opinion, that innovation needs to be where we put 80% of our effort, 80% of our funds, and 20% into flushing out developments with incremental improvements and knowledge. Um, it's right now it's very much inverted. In fact, in the US, with our federal government completely almost abandoning support for cancer research and drug discovery, we're at a point where we are looking at neither incremental improvements nor major innovation. But hopefully in the future that corrects itself, and hopefully, what we learn from that is that we need to push harder on innovative approaches, novel thinking, and put less of a impetus on those incremental. Just reverse it. It's not that those incremental improvements and those flushing out of ideas is bad. It's not. It's critical, but the balance is incorrect, in my opinion.
I always thought, I mean, if I find this point interesting, incremental improvements versus really breakthrough research, breakthrough innovation, where you said finding something new, innovative, tackling new problems that nobody thought about. And of course, it's risky. I always thought that basic research and science is the place for that, and deep tech startups are the place for that. And you experience it otherwise.
I think that startups are definitely a good place for that if you can fund it, which is also
Mm.
very tough to do. But that's the thing. Academic research has suffered such in the last 10, 15 years, the incredible pressure around funding. Everything has gotten more expensive. Buying reagents, paying people. When I was a graduate student, $25,000 was a reasonable salary. Now graduate students make $50,000 and their faculty mentors also have to pay their tuition, which is another $25,000. So when you get $1 million grant from the government, and that's kind of the biggest grant you get, an R1, you that money, and the university takes 40% of it, let's say, that money quickly disappears into staffing needs and into the high cost of reagents. So the money that is deployed to fund research uh, has not kept up with inflation, has not kept up with the increase in cost of everything. So each dollar is worth less and less, has become worth less and less over time. And so the competition for each dollar has become higher and higher. And in fact, I think the National Institutes of Health just ruled that, people can only submit 6 grants per year or something like that, because there were many people who were submitting 40, 50 grants a year. And so the competition has created this academic environment where you really can only afford to pursue something if it's almost guaranteed to provide you with information that you can predict. That's not a high risk, high reward situation. That's not a situation that promotes innovation. That's a situation that promotes incremental
Mm.
growth and improvement. And that really has been the fundamental problem. And there are certainly high risk, high reward mechanisms, and through the NIH and NCI in particular, um, directors awards and certain pilot grants and concept awards. But the overwhelming majority of funding went to incremental improvement, incremental knowledge. And to me, that's an inversion of, as you said, what academic what academics is supposed to be. And really, it's a financial problem. It's just come down to who's funding it and why, and what are the issues around that.
Mhm. Yeah, that's true. That's true. And then, I mean, I believe 9 out of 10 people would have said in your situation, yeah, I have a good idea, I know that it's good, but I don't do it. And the inventor of Spider-Man said in a commencement speech, uh, don't do it. If you have a idea where you think it's genuinely good, pursue it and don't drop it because other people say it. The interesting thing is, what motivated you then to pursue your idea and not say, okay, I do incremental research and that's it? What motivated you to bring your idea into a company?
Yeah, I think every cancer biologist faces this fork in the road. Am I a researcher, or do I want to cure cancer? And it turns out, I mean, they don't tell you that in cancer biology graduate school, but it turns out those are two very different jobs. Studying cancer, being a cancer researcher, and curing cancer are two totally different uh, career paths. And maybe I found that out kind of late, but it just, it becomes clear which one you are very quickly, and they're both incredibly important. I mean, would have I would have nothing to pursue if I hadn't been an academic researcher.
Mm.
And so the incubation of ideas and the support of ideas in the academic community is essential. That is really, and that's what's so horrible what's going on now in the U.S. That is our nest where ideas are hatched and where they extend from. For me, the limitation of that comfortable incubator, that comfortable nest, was that I couldn't feasibly cure cancer from that position. And that's really who I am. That's really what I care about, is the translation of the best possible idea to create the best possible drugs to treat as many people as I can. If when that's your motivation, you quickly feel hemmed in and restricted, uh, in your comfortable little environment, right? And so the desire to fledge and to leave the nest, and to make your mark and to truly have an impact, for me, that's the overwhelming motivation. Um, to actually put a drug in somebody's hand and see them take it or see it being infused and know that they're going to live a longer life because of all the hard work and the blood, sweat, and tears that went into that. That's the most important thing I'll ever do. That's my goal. For other people, the goals are different. The creation of a canon of knowledge, that's so important. The training of a next generation, critical. Those education missions and the creation of knowledge missions. But for me, what gets me up in the morning, what motivates me, is not really those things. And that's what I learned about myself. And when you learn that about yourself, you really don't have any choice. You are organically led to the decision where you have to get up and make the move.
Yeah, that's great. That's great. You mentioned earlier artificial intelligence, and your drive is to cure cancer. And this gets you up in the morning, and your goal is not to develop a better understanding of a thing. When you think about your goal, curing cancer, where do you see the best use for artificial intelligence and all these companies that are now currently try to understand artificial intelligence in drug development? What should they do, in your opinion, so that their tools are directed in curing patients?
Yeah. There's a lot of places, really. I think it's a tool. It's like, I mean, there's a lot of hype around it for whatever reason, because again, human behavior, we love that, right? We love that, to ride that wave of innovation and excitement. And we forget that everything has its limits, and a lot of it is it's certainly hype at the moment. But it is an incredible tool, and there's certain parts of the drug discovery process that are about processing massive amounts of information, and making predictions about them. So, for example, there are millions and millions and millions of small molecules in the world that we know what their toxicity profiles are. And an AI tool can train on that and develop really good understanding beyond human capacity of molecular features that are associated with toxicity. That is a really important place where we can use the AI tools to say, no, you're making a mistake. Don't spend 10 years on this molecule or $1 billion on this molecule because the tox profile is bad. It's wrong. You're going to hurt people. To know that up front based on, an AI's interpretation of millions of data points, that's super, super helpful. That's going to accelerate the process and prevent mistakes. But it's not a magic, it's not a magic wand. It can't create it's we're training these AIs based on the information that exists. So
Mm,
if your information is faulty, like, a lot of the scientific literature is faulty, there's issues. If you're training your AI, somebody once said to me when I was a very young student, garbage in, garbage out. And I thought it was a really harsh thing to say about the experiment that we were developing at that point. But I nevertheless think about it all the time and it holds up.
mm.
If you're training on data that's faulty or data that's incomplete, you will make mistakes and then the whole thing is pointless. But there are aspects of drug discovery where the data is complete, where there is enough information, and that's where AI can really, really be predictive and valuable. Because particularly in small molecules, where so much is known and you really can train really powerful systems to give you predictions, give you a subset of molecules to test. And instead of testing 2 million molecules over the course of 5 years and spending $10 million to do it, you can now test 100 molecules in a medium throughput assay, spend a couple of months doing it. You save time, you save money, you get better assets in the end. That's where AI is being directly applied and deployed really, really in a really, really useful way. Um, Whatever the hype is telling, that part, I can't speak to that, but there are definitely areas where it's going to be a really critical tool.
Yeah, the hype, there's a lot of storytelling. I think that the best story that I heard is in future, you will get personalized medicine, fully automated robots, and AI will
Robot,
develop
yeah.
new drugs in real time. Doesn't take 15 years, it takes a few days, and that's it.
It's an extreme example, but I think could we do it in a year or two once these, at some point when these tools are really sophisticated? Yeah, I think they will and are accelerating drug development to the point where that will be possible. And I think that there is value in that because if you develop a personalized medicine profile for someone, that same profile is going to be useful for a million other people down the road. So I think there are certainly people out there who really do want to flip that script and say, instead of studying populations and coming up with drugs that we think are going to impact and be useful for a population of people, let's start with one patient. Let's cure one patient and create a drug or a therapeutic strategy for that patient, and then deploy that strategy for everybody who comes after that fits the certain parameters. It's a completely inverted strategy
Mm.
from what we have all been trained to do. I think it's fascinating. I think that it's very resource dependent. What's scary is the idea that billionaires and millionaires will be able to afford those pipelines for themselves, and others won't. And so we have an even vaster separation of have and have nots that results not just in quality of life, but life itself. That's a disturbing possibility. But at the same time, for people who have resources to develop those pipelines that then benefit everybody, there's value there. So it's a little ethically complex, but ultimately, perhaps the ends justify the means in that case, perhaps.
Is it really an ethical problem?
Only when someone is asking you to do it. The idea that a billionaire community could develop therapeutics to to extend their own lives, and for example, if taxpayer money is going into funding a company, partially that is doing that kind of thing, that's complex, right? You're
Mm.
spending the taxpayers' money to develop a therapeutic approach that benefits first one person. But then as long as you can deploy that for everyone else in a beneficial way, I think it's a rational approach. It's an inversion of how we've always done things, but it's certainly a rational approach. But I see where given the moment that we're in, socially, and this widening gap of socioeconomic status, I can see a world where billionaires developing their own cancer cures while average socioeconomic people are dying it's a complex picture for those reasons.
But similar to your story, where you said you saw this inflection and you saw this problem in the academic environment, and this got you moving into another direction. But isn't it the same with this problem that you described? I always thought innovation is basically, I mean, in my opinion, innovation is driven by single people,
Yeah.
not by politics, not by governments, not by philanthropic entities. It's usually one person who says, this is wrong. And I try it. And even if I look dumb, I believe in it. And many fail with that. And I always think this zest of billionaires, I mean, they are now in their 50s and 60s and 70s and they're afraid of death. Uh, at the end of the day, what happened when I look at other industries is one person took the leap and invested in an area where everybody said, that's nuts. You are crazy. This never work. Airplanes developed that way, for example. I mean, without people risking their lives, and many died in this process, we would not fly today between Europe and the United States as everybody can do. And I always thought that there is a proliferation of knowledge. So when one person can solve it, like running a marathon, running 1 mile under 5 minutes, I think was a landmark, if I remember it right. Other people can copy it, and they will copy it, and then it just disseminates. But I think there will always be this gap, so that some move first, a lot fail, and then others benefit from it. Do you really? Why is this a problem in this
I think
context?
the line in the sand is when it comes to life
Mm.
and death. Um, Wealthy people being able to fly and less wealthy people not being able to fly is not life and death. When it comes to curing disease, we have a responsibility to make that available. That's a public endeavor, and that's why it's always been funded by governmental resources. Um, and so there is that danger of misinterpretation. Ultimately, the goal is to provide a life extension pipeline to everyone. Um, and if you start with very wealthy people because they can afford to pursue it, I don't think there's anything inherently wrong with it. But I think it's a fine line when you're talking specifically about life extension. Just, development of technologies, the radio, television was first only available to wealthy people, right? All of those things. And people aspired to become wealthy so they could have those things. And that's also normal human behavior. But when you're talking about life and death, you create a different paradigm, right? Life and death should be equally available, even though it's not. We all know that wealth extends life and poverty shortens it. Um, and it's you don't want to add to that widening gap, I think is the danger. And it just has to be it has to be dealt with nuance and carefully, because it is an opportunity. We are at a precipice where very wealthy, uber wealthy people can improve their own health. Um, and as long as that's done in a way that benefits everyone, I don't personally see a problem.
Mm.
If there is at any point an effort to limit the application to wealthy people, that's where we're going to find, that's where you cross the line, in my opinion.
I think it's not possible in Europe at the end of the day. So, but when I think about drug development, I mean, proving that a drug is safe short term, it's a great thing. But you always have this long-term effect, so would not be too worried so much. Let some people walk first, and then see what happens in 5, 6, 7, 8, 9 years down the road. And when it's safe, at the
Yeah.
end of the day, I think then is the right time to really push it out into the public. It's not so much, from my opinion, holding it back with bad intent to withhold it from people and not give them the opportunity. In my opinion, I think it should be a cautious step before we really put it out to 8 billion people on the planet or 9 billion people. We must be sure that this thing is really safe.
And I think the framing of uber wealthy guinea pigs is fine. I don't think I would object to that.
Yeah, I got now they hear it, or we have recorded it. But let's go back to cancer research and to your company and your role as a CEO. I mean, there is a lot of hype about AI, and you found your calling with your company, and then you get into this hype bubble where a new thing comes up. What helps you to stay focused on your mission and not jump ship and say, okay, let's move on to the next thing?
I know we've had these conversations with potential investors so many times where you know that if you just say something hypey around AI, somebody will write you a check. And, it's just not who we are as scientists. If we're scientist founders, we're technical founders. And our first obligation is to the truth and to practicality and what's really doable. And I think that my co-founder, Ann Carpenter, she's a world-renowned machine learning AI expert from the inception of the field in focusing on drug discovery and image-based drug discovery. She definitely keeps me grounded. She understands what's really practical, what's real on a fundamental technological level, and is the first person to say, when we meet with someone who's for example trying to sell us a service that's very hypey, that's very AI driven, she'll be the first one to say, this is no different than any kind of open source software you could find, or here's the difference, here's the unique feature. Very practical, very grounded, and so she doesn't let us get carried away into that world, because she's a really a tentpole person in that community, and so it's very, very connected to what's real and what's not. And it's just a really interesting time whenever there's sort of a hypey innovation, and a million companies are formed to take the money that's being offered to develop it. Um, you know, it, it- you have to be very careful. It's a good way to fail, right? By selling hype and not really having the practical way to deploy it, to deploy the technology, and that's something that we stay very far away from. Anytime there's a bandwagon of people around anything, to me that there's like a red flag that goes off, uh, right above that, and I run the other direction.
I mean, currently I have a lot of had some speakers on my podcast and live show talking about AI. They have companies developing AI, and they always have a little bit of a challenge when it comes to application in drug discovery. For a simple reason. So when I think about this process, you discover a drug, bring it through preclinics and then into the clinics. I mean, ideally, what you want is just advancing those molecules or drug candidates to generally speaking, uh, that are safe and effective. And ideally, you know it from day one and don't have to do a lot of expensive studies with that and then can directly go to the patient. I think there are two mistakes that can be made. One is selecting the wrong drug. So you bet a lot of money on the wrong drug and find out in phase two it doesn't work. But the other one is also critical, not selecting the right drug. And when I now look at the claims of AI companies where they say, I can predict the safety profile and the efficacy profile before you make any study. How can we be sure that this really works? I mean, isn't it like a lottery predicting the numbers?
Yeah, it's like any prediction. It's just a prediction.
Mhm.
Um, I think what we want to do and what we are able to do with those tools is to reduce the number of molecules that we need to test.
Mhm.
Some are going to be obviously bad to the AI based on millions of profiles. That's great, right? If that takes 50% of the molecules that you would otherwise have been testing and wasting time and money on, if that throws them in the trash at the outset, that's great, right? You've now focused down on a subset that you can work with. Ideally, the tools are so precise that, let's say you're only dealing with the top 20%, right? You're able to discard 80% of your potential molecules early on because of toxicity profile or metabolic profile. Now you're testing the prediction. You're saying, I know that I have my top 20% of molecules. Let's say it's 500 molecules. This is what I'm going to deploy my time and effort on, right? I just saved myself a ton of time and a ton of money. It doesn't mean that what the AI predicted is the best compound is ultimately going to be the best compound. And I don't care. All I want to do is reduce my wasted effort and my wasted money, right? I want to focus on a set of compounds, a set of drugs that have the best properties. And if the AI tool can help me do that, I win.
But what,
But the rest is hype.
I mean, when we talk about future, so I spent a lot of my life in planning companies and come from that part. And I liked in the 90s when I was at university, I liked the systemic thinking. So also seeing companies like an organism. And when I think about established businesses, what we want is driving the rate of mistakes and errors and failures down to zero and optimize the process, every single part of the process to really get the best product to the customer very fast and very quickly with minimized failure potential in that. When I think about innovation, I think driving down the failure rate and the rate of mistakes and things that doesn't work risks that we miss the target entirely because it's just applying a tool set in an environment where it's not appropriate. I always thought innovation needs failure so that we experiment enough, try enough new things, and make sure that we find new ideas to do new things better that nobody thought before. How do you see that? When we really try to reduce with AI this failure rate and say we focus just on a few parts, isn't that risking a little bit serendipity? Might we not drop things that might work at the end of the day if we just think a little bit harder?
That is the risk, right? That's the risk with these tools is that we miss something. And I don't know what the answer to that is. I know that what we're doing now to avoid that mistake, high throughput screening and very reductionist, very simplistic approaches, screening millions of molecules without any kind of funnel or filter to give us starting points for better material. That has is too expensive. It's compromising the industry. It's allowing the process to slow down to the point where international competitors can beat domestic competitors by copycatting because the process takes too long. Um, for us, takes too long. So I think that a faster approach is better economically. It's better for patients. Uh, Fine-tuning our starting matter is a rational decision. Uh, Wide, unfiltered screening is the other extreme. It's too extreme. So on the one hand, you have AI that tells you, here's your only test this one compound. This is the best compound. Don't think about anything else. And on the other extreme, you have high throughput screening of millions of compounds, and they're both far too extreme. And what you want is a very rational middle ground where you're starting with enough compounds to cover your bet. And those compounds have good properties when it comes to toxicity and those things so that you're not wasting time and money. That's the goalpost for me when I look at these tools. Um, that neither of these extremes are going to be have been success stories. The way forward is that middle ground, in my opinion.
Yeah, I totally agree. I totally agree. And this is, I think, the tension that's interesting for entrepreneurs. At the end of the day, you need some focus to move a company forward. But on the other hand, you also need to be open to new ideas that you're not getting too narrow and missing then the next big thing.
Yeah, that's the challenge, right? I was
Yeah.
having this conversation yesterday. When are you holding too tightly to the original strategy? Uh, when to pivot, when to expand? Those are the toughest leadership decisions there are, and really only the CEO can make them. And you're out on that limb all by yourself, and it's pretty scary.
Did you find the golden rule yet
I
that
try
guides you?
I try to lead by consensus.
Mm.
I try to make sure that the big decisions, the key stakeholders are all in agreement. But there are definitely times where I feel like Mama knows best. And I have to sort of put my foot down and say, no, this is the approach, even if people disagree. But I try to have that be a minor occurrence and have the vast majority of our decisions be consensus decisions, because I feel like without everybody's buy-in, we achieve nothing.
Mm.
For in terms of key stakeholders. So um, I don't think that dictatorship in a situation like this is particularly beneficial. I don't think it's beneficial in any situation, but particularly in an early stage startup, making sure that there's buy-in from everyone that is critical. That's my mantra. That's my golden rule of leadership.
Mhm. Yeah, that definitely works very well in these days. I mean, this focus and being a little bit more creative is an interesting point also when we look at cancer research. I think when I started, the first thing that I realized in 2006 was basically mostly small molecules, and then there was a phase where everybody were talking about antibodies. Small molecules were a thing of the past. Then I think we had ADCs.
Mm-hmm.
Then with 2020, mRNA appeared on the scene. And when we look at policymakers, especially here in Europe, the United States, some are entrepreneurs. I think the US president currently has an entrepreneurial spirit, and they always risk being too focused and just betting on one thing and eliminating all the others. What's your opinion? I mean, when we look at the landscape around solid tumors, should we focus on one of these therapies, or should we go broadly and be more creative and say, okay, let's just try it all and see what works in combination?
I think that the trouble with large molecules from a purely scientific perspective is they're bulky. And the
Mhm.
solid tumor, they have trouble entering the solid tumor space, and they have trouble persisting in that space. And I think that as first line therapies, that's a real problem. If you can't enter the field of battle, you're not going to win. Um, and so I think small molecules win in that space because not only are they solid tumor permeable, they're cancer cell permeable, they're immune cell permeable. So I think that, yes, is it an older approach? Yes. Is there a reason for that? That it persists? Yes. I see the beauty of the innovations and I see where the excitement is, but the only way that those large molecules can really do the job is if we alter the tumor ecosystem to allow them to get in and to function. And so we take a sort of two-pronged approach there. Our goal is to get small molecules into that solid tumor environment to soften up that ecosystem so that large molecules can penetrate checkpoint therapies, for example, even cell therapies. But the solid tumor microenvironment as it stands is a fortress, right? It's an absolute fortress, and those larger molecule drugs have a real hard time penetrating into that environment. mRNA is interesting. mRNA vaccines are an interesting approach, but just like immunotherapy, just like cell therapy, they rely on an active immune system. And the overarching problem with the tumor ecosystem itself is that it's immunosuppressive. And the best cell therapy in the world, the best checkpoint inhibitor in the world, cannot overcome that immunosuppressive nature of the solid tumor. So our job at SyzOnc is to get in there with small molecules, rewrite that script so that the solid tumor, to weaken it, to create essentially portals, and in an environment where large molecules, cell therapy, antibodies can be successful.
Why is that environment so tough to overcome?
And this really gets to the crux of what we're doing. All cancer biologists are familiar with the words extracellular matrix. They all understand that there's more in the tumor. Fundamentally, we all understand there's more in the tumor than just cancer cells and immune cells. But in terms of practice and application and drug discovery, those ideas, again, they're too complex. It goes out the window. Actually, there's a great story. I remember talking to a protein chemist who does mass spec, and he said to me, these very matrix-rich solid tumors, when we're doing mass spec on them, we just separate the cancer cells from the other stuff and throw the other stuff out. And I said, Paul, the other stuff is the problem. That's what we need to study. That you're tossing out the the critical. And he said, I know, but it's too complex. We don't know how to work with it. And so that complexity, that tumor infrastructure of extracellular proteins, that signaling network, that's the problem. It's the problem we have to deal with for any of our other therapies to be successful. And it's just been kind of systematically overlooked and ignored uh, by everybody's pharma pipeline because of its complexity. And that's where we come in. So we've developed tools and systems that allow us to evaluate uh, how and modulate that environment. And that's really what's been lacking in the field is a good representation of that solid tumor immunosuppressive infrastructure. And it's turning off T cells. It's like the patient's own immune system can't get in there. Cell therapies can't get in there. It's a complete black zone. It's like imagine the tumor, it's like your Wi-Fi signal gets turned off. There's no connection between the immune system and the tumor. Um, the tumor just turns it off. It's just, sending signals to the T cells that says, nope, go away or go to sleep or you're tired. You can't be here. And that is an overwhelmingly powerful environment when faced with the therapies that we have now. So our approach is to get in there, turn on the lights, turn off that immunosuppressive feature of the microenvironment, and then we can start talking about large molecules, antibodies, cells that can get in there, supercharge the immune system and take care of those tumors without necessarily harmful, having to do all the chemo and radiation and all of that. It's that immunosuppressive component of the microenvironment of solid tumors that that's the future. That's the final frontier. That's what we've all been avoiding because we don't know what to do about it, and have been overwhelmed by the complexity of it. And that is absolutely where we need to pour our resources and time and energy into mitigating that problem. And then, we have this arsenal of tools that would be much more successful.
That's an interesting point. I mean, while you were speaking, this concept of the Berlin Wall popped up in my mind. It was the concept of 1989 when I was a kid. So it was the huge event in Europe when the Berlin Wall fell. And
Mhm.
basically, what tumors are doing is like being a Berlin Wall. So you can't do anything on the east side of Berlin
Mhm.
without overcoming this wall. So you can have the best tools to build houses over there, but you must first get over the wall and
Yeah.
negotiate with the other side so that they let you in. And isn't that obvious?
Yeah.
Uh, but why don't we have a solution for that yet?
It's hard. It's just been very hard.
Mm,
There's some great experts in the field, people like Richard O'Hines at MIT and Ellen Pure at UPenn, that have been pioneers in this field. Um, and it's been a very complex problem to solve. And I think we're there now. I think we understand enough about that tumor infrastructure to start messing with it and to start weakening it. Um, but it's also a feature of the tumor that's unique to any given tumor. So, for example, that matrix component is made up usually of individual collagens,
mm,
different types of collagens that are very heavily modified by different enzymes. And so, in sarcomas, the type of collagen that is the bad actor, that it truly is just the devil, is collagen 6, collagen type 6. In pancreatic cancer, it's collagen type 1. Uh, but collagen type 1 in sarcomas is actually protective. It's a tumor suppressor. So these are very complicated biological pathways, and the regulatory pathways around them have simply been it's been a niche. It's been a niche area for people like me, for people like Ellen, for people like Richard, who kind of been ahead of the curve and know that this is the critical problem that we have to solve. Um, and the complexity has not been completely revealed yet. We're working on that. Of course, all of us are, but it's complicated. It's been a hard problem to solve. And what we need now are small molecules that will get control of that system, and essentially act as Trojan horses and allow the army behind it of immune cells, of cell therapy, of other drugs to get in and do the job. This is the problem of pancreatic cancer, of glioblastoma, of soft tissue sarcomas, of liver cancers. all the dreaded diagnoses, all the really hard to treat solid tumors, they all have this central problem of an immunosuppressive microenvironment that is a fortress made of a dense, heavily modified protein structure that no one has been successful or really tried to translate into the clinic. And we are among the first to do this, to finally say, we've got a handle on this complexity. This is what we're going after. We're going after this fortress. And then when we're done, everything else can flood in and do its job.
So, in principle, if I understood you right, your goal with your company is really focusing not only on understanding this fortress, but taking the defenses completely down,
Right.
tearing it down for all potential cancer types that we have in the world. And this is always a different problem. It's not the same problem for every cancer. It's really that it's unique.
Same problem, different players, different molecular players. And that I think that's one of the things that's made it so complex. When you talk about proliferation pathways, all cancer cells use the same proliferation pathways,
Mhm.
roughly. If you're going to inhibit MAP kinase or RAS, that's a universal truth. When it comes to this complex defensive mechanism, each solid tumor type is relying on essentially their background tissue to decide how to build the fortress. So in a muscle tumor, collagen 6, that's a normal muscle protein and essentially is being hijacked to form the defensive barrier. In pancreas cancer, collagen 1 seems to be the protein that's being hijacked to create the defensive barrier. So the first step really has been to characterize which are the critical proteins, the critical ecosystem proteins in each of these tumors, and then develop a strategy to target them. You can't really target them head on, in my opinion, because they are critical normal proteins. So, you don't, if you take out somebody's collagen, they turn into a pile of goo. Like it's not an ideal approach. But in each case, there's going to be enzymatic regulators of the defensive proteins and that are unique to the tumor, that are overexpressed in the tumor, that are specifically upregulated there, that we can go after. And that's our goal, is to hit those regulators of the defensive pathway.
That's amazing. So if that works, it means that everyone with a therapy needs your support at the end of the day.
That's what I like to say. And
Yeah.
that's been my approach, for the last 2 years is we're just trying to help you. This is a unique situation in therapeutics where we are not trying to replace you at all. We're not trying to say that all these other therapies are critical. Each individual patient population, some of them are going to work, some of them aren't. But in every single case, those environmental defenses have to come down for success. So for us, we believe in that as has monotherapy potential because the patient's own immune cells will also be able to access the tumor, and that may be sufficient for some people. But in the cases of more advanced disease where their patient's own immune cells aren't sufficient, then you have an arsenal of cell therapies, immune checkpoint inhibitors, and so on, and potentially mRNA vaccines as well, that can really, really come in and clean up and finish the job. But before anything, it's like we've been lobbing missiles at a fortress, right? And we're not having the impact that we want. And we're all sitting around going, how come we're not, why isn't it working? Those defenses have to come down first,
Yeah, it makes sense. I mean, from the military point of view, it's easy to understand them and first like take the defenses down and then send in the infantry.
right? It makes a lot more sense than just sending in the infantry just to bang its head against the wall, right?
But the current approach in the industry is to just send in a lot of infantry, no matter what happens.
No. Yeah, I mean that's where we are now.
Mm.
And I think, and again, it's not that people don't understand this issue. They do. They simply have not known how to deal with it. And I think, and that's where we come in, is those tools to mitigate this issue. But yes, absolutely, it's well understood. This is the problem when it comes to cell therapy, CAR T therapy, and solid tumors. This is the problem when it comes to checkpoint inhibitors. Those T cells cannot persist, remain active in an environment that has these defenses. And the T cell engineers' response has always been go back to the T cell engineer a better T cell. We just constantly trying to engineer a better T cell. And while I respect that, as a cancer biologist, I'm saying, guys, you're just trying to make a better missile, right? But these defenses are impenetrable. So we have to bring down the defenses, then your missiles are fine, right? But first, that wall has to come down. First, this dome of impenetrable
Mm.
protein defense has to be dealt with.
Bigger missiles don't always work. I mean, when I think about the patient, most cancer therapies are toxic, highly toxic. And at the end of the day, it ruins the body of the patient. I mean, it's great. The cancer is in control, but the patient is dying from something else. And when I understood your approach right, when you take down the defenses, you need much less toxic material. And this is beneficial for the patient long term because it doesn't destroy the patient's body at the end of the day
Yeah,
as much as the old way.
100%. Our goal, and this is the difference between focusing specifically on the cancer cell, just on the rocket fuel.
Mhm.
Um, I don't care as much about hitting the cancer cell as I do about exposing the ecosystem to our therapies. Because the cancer cell will always get smarter, it will always evolve. We've learned this over and over again, and it takes more and more toxic drugs every time to try to hit it, and you always end up with the same sort of recurrent problem that needs invasive surgeries and really, really, as you said, sometimes the cure is worse than the disease. And so my goal really has been to leave that strategy in the past and focus on this tumor ecosystem, on bringing down these defenses. And that is a non-toxic prospect, or at least a very lower toxicity. The type of enzymes that we want to hit, they're only going to be expressed in these unique conditions of the tumor. And so we get them, and the rest of the body should be unaffected or very minimally affected with the right toxicity profile for the small molecule, which is critical. That's the dream, that we develop these molecules, they don't harm the individual, they only bring down the defenses on the tumor, and then the vast arsenal of tools that we have can do their jobs.
So, from what I understood from our conversation so far, is that it's a new approach in the scientific community and it's game changing in that area. But to bring it to fruition and into a product, you probably need also the support from the pharma industry and from investors. So, you need capital to develop the solution, and then you need the buy in from pharma to bring the solution to the patient and combine it with other therapies. How is the reception in the investment and pharma community of your approach?
Cautious.
Why?
The again, complexity, the sort of the Me Too-ness of everything that we do. And I told you the story that I heard recently about a woman who was working in a technical role, and she went to a new company and was asked to validate a target, and she said, I validated this target for 7 different companies. Everyone has been chasing the same uh, targets, and that's problematic, obviously. But I think for pharma companies, risk aversion and you know committing their massive companies, they're responsible for a lot of lives and a lot of livelihoods and a lot of shareholders, and that's a big responsibility. And that responsibility can really suppress innovation, can really suppress that high risk, high reward attitude. And I think it's a completely understandable place to be. And I think we've seen caution as a response to what we're doing, but also excitement. It's a I really want to see what you can do with this. I support you. I'm excited about what you're doing. I really, you know, but cautious about deploying capital around it in a tough market, right? And so the kind of investors and groups that we've had a lot of traction with are people who don't have those responsibilities. So foundations with venture arms, American Cancer Society, Cancer Research UK, these are great organizations that their obligation is simply to move the field forward and to be catalysts for more funding and for innovation. And they've been really great supporters for us. And that's where we focused our efforts, really is on mission-focused investors who see the value of what we're doing, see its potential, and don't have that risk aversion, um, don't have such incredible responsibility to shareholders and uh, to others that they can afford to take risks.
That's an interesting observation. I mean, when I think about investment styles like Warren Buffett, it's clear that this is not for him. But when I read the literature about venture capital in the United States, venture capitalists always are portrayed as seeking asymmetric bets. So high risk, but also high reward. So when you succeed, you make them billionaires, and there is, of course, also high risk of failure that it doesn't work and you can't overcome the complexity. I always thought it must be perfect for US-based VCs, and why it's not. And it doesn't sound like you're convinced that they throw
I
money at
think,
you.
as many people have said to us, oh, if you were raising this in 2022, people would be throwing money at you
Mhm.
and there would, be no problem. And I think part of what is happening is simply market forces and a lot of people sitting on the sidelines waiting to see
Mhm.
what's going to happen next. And I think, there's nothing we can do about that timing. It's not really mattered for us very much. We're very happy with mission focused investors, and those kinds of, you know, people who are as focused as we are on our ultimate goal, which is curing cancer, for these early round, we're an early stage company. When it comes to Series A, I think, we'll have be able to show the de-risking that we've de-risked the strategy and we'll have assets. And I think all those, hopefully those venture capitalists will get off the sidelines and be happy to engage at that point. And I think that traditionally is what happens other than in really crazy boom markets, that they're cautious with their investments, and I think that that's fine. I think that this type of environment creates really strong, robust companies that are extremely practical and they're not hype. They're not going to die in 2 years. Surviving this kind of these market pressures makes you really strong, and really, really robust and flexible. So I think it's actually a really great time to be coming up, because it's teaching us how to be cash efficient. It's teaching us how to be innovative without breaking the bank. We're getting the best possible training. It's almost like being a PhD student, where you're just, we're coming up as strong as we possibly can because of these market forces. And I wouldn't have it any other way, to be honest. I think this is the best possible training for somebody like me, for deep tech founders. You know, being in a market where everybody throws money at you and there's, I'm sure that's lovely. I wouldn't know, but you don't get the training that we're getting going through this environment.
I agree. Funding is down to 2012 levels currently, so we are, I think, at the worst. So it should get better, and the best companies always start in hard times, not in good times.
Isn't that the case, right? And so I feel
Statistics.
like, yeah, I feel like that's when I'm worried about it, when I'm having a tough day thinking about that, that's what I tell myself, right? Is that the best companies start in these hard times. And that's what we want to be. We don't want to be fly by night. We're not looking to exit in a year. We're looking to cure cancer, and that's a long-term play. And we want to be as robust as possible doing it. And we want partners who share that mission. And there's many VCs out there that have a mission that is for the benefit of humanity, right? That they want to invest in things that they can be proud of. And I think that when the time is right, those are going to make great partners for us.
Absolutely. When we think about the great companies that started in crisis, if, I saw a video about the first company of Sam Altman. So Sam Altman started in a crisis. Now look at OpenAI,
It makes you
Uber.
stronger. It makes you smarter. It makes you, yeah, it makes you better.
Yeah, that's true. Where are you in the development with your company?
In terms of fundraising or in terms of operation.
Both, both operations, fundraising, and development of your products.
Yeah. So we've been having a great time building relationships over the last year we raised a pre-seed round, and that has funded operations for the last 18 months or so. And we've been laying the groundwork for this seed round. And I think it's coming to a head in the next couple of months. I'll be extremely proud to close that round in this environment. I think that's, going to be one of the proudest accomplishments of my life. And I'm really, really excited about the partners that we have that really care about what we're doing. And that to me, that means so much to partner with people who share our mission and for obsession, for lack of a better word. Um, and then in terms of building the company, this is a really interesting time of lean and mean companies where, a handful of people can achieve a great deal by using all the external organizations, partial, fractional contracting, and all of that. And I think that we've been successful using that model. I don't imagine us growing to be a 500-person company in the next 18 months and deploying all that capital. I imagine us continuing to stay lean and mean and take advantage of all of those systems that have been put in place by these incredible service companies that are provide any possible service you could imagine at the highest level, incredibly, quick and accurate. And I think that that model is a really exciting one, is one that we're going to pursue, at least in the short term. I can imagine us being a 30-person company. That's my dream. We're a 30-person company that has fractional employees globally, that's using every resource available to us, that's incredibly cash efficient. And the full-time employees that we have are brilliant innovators and support staff that are just, each one is exactly what we need, doing the exact role that they were born to play. And, no excess really anywhere. And just being the most innovative and productive team that's not burdened by regulation and overly hierarchical, big company problems, people who spend 99% of their lives in meetings. And so work 90-hour weeks and can't accomplish anything because they spend all their time, discussing it instead of doing it. Um, I really hope to leave that model in the past and not become that. For especially when you're working with scientists who measure the achievements and the success of their life by the things they're able to do, not the things that they say, not the hype, not the marketing. Um, Preserving a corporate culture where they can do that, where they can actually be functional 90% of the time, that's really the dream.
Yeah, yeah, completely agree. When we look at gender in biotech, and to switch gears from cancer a little bit on women in biotech, how do you see the situation in 2025?
Yeah, it's interesting. I'll tell you that I went to my first JP Morgan week in San Francisco this past January, and I saw two things. And I think that JPM is kind of representative of the industry. When you go out there, you really see what's happening. And I saw two things. I saw women supporting women. I saw groups of women uh, meeting and dinners, and there was really a women in STEM support network that was evolving, but it was incredibly small. And I would say that 1% of the people in San Francisco that JPM week were women. And very stunningly, I was with two female colleagues and we were having dinner in a restaurant that week. And you walk into the restaurant and there were 500 men in that restaurant, all wearing the same blue suit and the same white shirt and the hair gel, the exact same hair gel. And it was like a clonality event, like really, it was stunning, stunning. Tables of 25 people having, working meetings, biotech meetings, and there wasn't a single woman at the table, or at most one. It was a stunning picture of where we are as a biotech community. And I'm no expert in this. I'm not, but I'm an observer. And I certainly saw that the women who were there very much wanted to support each other and wanted to grow their presence in the biotech community, but it's still a very, very underrepresented group.
Why is that, in your opinion?
Um, Observationally and anecdotally, I think the community of biotech come out of a few very high profile institutions, a few very high profile business networks that you know has been a boys club for generations. And I think women are just now kind of starting to break into those circles. And I see it every day, really, really powerhouse women who are founders, who are serial founders, who are now VCs. I'm talking to more and more women in VC calls and in fundraising calls, and that's been great. So I hopefully it's at a tipping point where it becomes more balanced. But I think just historically, these institutions, these networks, these fraternities have been the domain of men. I don't think it's necessarily I think it's just sort of, again, natural human behavior that like attracts like. And if you're you're a Harvard Business School grad and you have your network of other Harvard Business School grads and you decide to start a company together or you decide to start a fund together, you're doing that with your friends. And it's, again, that sort of natural human bottleneck issue. But I think more and more women are breaking into it. And I think that the future is bright on that score. I see it as something that is slowly changing. And I don't think there's anyone to blame that anyone is excluding anyone else. It's simply, again, just human behavior that is
Yeah.
hopefully changing, that is becoming more inclusive, just because if more women are in the space, more women are interacting, more women become part of these groups. They establish the network, they become serial founders, they become investors. It's just being in the space is the first step, and I think that is changing.
So let me ask you the next question. What will be better with more women in biotech at JP Morgan? So when we say we bring it to 50-50, it's not 1% anymore. We plan for a future where half of it at least are women. What will be different in the industry?
I think maybe nothing. Um, that's a possibility. What I hope is that opportunity will become more evenly distributed. So I think something like 3% of all VC capital that's deployed goes to female founders. That's not a number that reflects the quality of female founded companies. That's a number that reflects what investors are comfortable with, right? Men are, going to naturally be comfortable with male founders, and perhaps struggle more with people they relate less to because it's all relationship. That's what I really found about fundraising and investing is that it's about building relationships. And I think sometimes it's tougher for men and women to build those kinds of collegial relationships together. I think it's perhaps easier for women to build those relationships with other women and men to build those relationships with other men, speak the same language and have the same kind of world experience and worldview. And so I think that number of essentially men only or majority investing in men, that's a problem that can be solved by having more women at the table, right? Then the comfort level will be, hey, yes, it's a female founded company. You can be okay with that. That's, don't be scared. And I think having women in those VC meetings and at the table will have a positive impact on that. There's no reason why 95 or 97% of VC capital should go to male founded companies. There's plenty of female founders out there doing amazing things, who are restricted essentially to raising money from other women or from funds that specifically set aside money for women. That's a bias that simply exists and should be combated for the benefit of everybody. We should be funding the best companies, not the companies that have founders that we can feel like we can go to the gym with, right? And
Mm.
essentially and I've heard that so many times from investors that'll say, yeah, me and this other investor, we did CrossFit this morning. And it's, those things that they have in common that create those relationships. And then they invest in companies together and they invest in companies where the founder is also a guy who wants to run Ironman marathons with them.
Mm.
It's that kind of relationship building that leads to investments. And I think that there's a very natural bias there, a very natural separation that we just need to work to overcome.
Mm. No, I totally agree. I think the only decision point should be getting the best minds into the industry, and JP Morgan is basically the Formula One of biotech and pharma, where the best should meet to make the best therapies happening, and not thinking too much about gender and ethnicity, and whether you can go with this or that person to the gym or not.
Yeah.
I think that misses the mark. Today, I listened to the All In podcast, and there is this All In summit currently, and Jensen Huang and Lisa Su from AMD and Jensen from NVIDIA were talking about bringing the best minds to the United States at the end of the day, and I think this should be the spirit.
Yeah, I agree. Again, it's a human behavior, human psychology bottleneck. Uh, it's not necessarily anyone's fault. It's just something we work to overcome, and having more women in the space, more women represented is the best way to overcome that bias and get everybody comfortable that we can overcome it.
You seem to have a good understanding of human psychology. Is this bias really so restrictive that people tend unconsciously to select certain people in and certain people out?
I think the statistics tell us that that's true. When you obviously when you meet somebody on a one-on-one level, you don't always see it, or you almost never see it. But then when it comes to job searches and when it comes to investment, the statistics, those numbers don't lie, right? 97% to 3%, that's a reality. So I like to think that and I do think it's unintentional. I don't think that there's a cabal of Stanford, Harvard men out there keeping women out of the biotech sector. I mean, that to me
Oh
seems
boy.
absurd. It seems a lot more likely to me that friends invest in one another's companies. Those are stories
Mm.
you hear over and over. That the field that this business is about relationships. And there are just natural inclinations when it comes to relationship building. That to me seems a much more realistic and a much more solvable problem. Then there's a cabal of systemic exclusion going on.
How would the society look like? The secret society of white old men, so,
Exactly. I
some in
mean,
Swiss.
in America, we call that the US Senate, I guess. But I don't think, I don't, it's just not, to me, it's not very feasible. To me, it's much more feasible that this is an overcomable problem, that it is just a natural product uh, of human psyche and of our higher education system and our networking system where, you know, guys hanging out at a bar, agree on, yeah, let's do this, let's invest. And,
I
you know,
think women do that too.
and
So
women
it's
do that too. Exactly. And so there's this separation. And the likelihood that we're going to solve that particular problem is low. And so I think just having balance in the representation in biotech and VCs and founders will naturally kind of solve that problem. Even if people tend to still self-segregate, everyone will be represented, and so it won't matter that much. But right now, it's a very stunning disparity.
I think, I mean, for coming from the business side, diversity is always good for decision making, and the ideas just get better. Different people see different things, different social backgrounds have different filters. Men, women have different filters, different ethnicities have different filters, and when you bring it together and manage, and this is, I think, the hard thing. Uh, we tend always to fall apart in groups, and women talk with women, and men talk with men,
Yeah,
and white people with
100%.
white people, black people with black people. But when you, and this is the leader's job, when you bring diversity together in a team and make people collaborate, that's when the real magic happens.
Yeah. And I think what's so funny about science is science has no gender, science has no race. We work with people of every possible creed, background, color, religion, everything. And what matters is overlapping interests and ideas, and are we passionate about the same thing? And I think Perhaps that's why I've always been so comfortable in science. That's why it's always felt so natural, because no one cares about anything but your ideas, the quality of your ideas, and how well you defend them, and how good your data is. I mean, it's ultimately merit based in that way. But like all reward systems, of course, there's bias in it as well. But when it comes to just the pure science of just being in a room with scientists, it's, you only are thinking along their scientific contributions. You just don't care about anything else. And that's always a fun place to be.
Yeah, that's true. Karin, help us spell your name right. I'm from Austria, and Karin is a typical Austrian name. How
Mhm.
do you spell it in the United States?
It's the same. My paternal grandparents were from Vienna.
Mhm. Oh, great. Oh, great.
So yeah,
Yeah.
that's the it's a the Austrian connection is real.
Okay, so this is good. Time flies when you have fun. We are almost 2 hours in our conversation.
Oh goodness.
How about spending the last part looking towards the future?
Yeah,
What
absolutely.
what's your vision for solid tumor therapy? How should in 10 years when we look at the point 10 years into the future, how will it look like?
Yeah. The perfect vision that I have is really when a person is diagnosed, all of, the incredible tools are applied, the profiling is done, the ecosystem is defined, the right small molecule goes in to bring down defenses, the cell therapy goes in or the checkpoint inhibitor goes in, and that person has a normal lifespan. That's the dream. And I think it's a completely feasible strategy uh, for 10 years from now, given what we're doing. And I think that the more people consider that view, the more people will say, like you did, isn't that obvious? Isn't it obvious? And it is. We simply have to have the conviction and the will and the money to where our mouth is to get it done. But that's my vision of the patient journey as a much less scary, a much less destructive, painful process where information is key, it's gathered quickly, the tools are present, and everybody goes home at the end of the day to their kids and doesn't live in fear.
Yeah, this would be great. Is this your boldest vision?
That's my bold vision. Yes.
Yeah, yeah.
Yeah.
When we look at today and you had the possibility to allocate the resources in the industry to make that vision happen in 10 years, where would you put the money?
I think best biology, new biology is going to win. We're not going to win on chemistry. We're not going to win on, uh, you know, the most aggressive antibody. We're going to win on the best targets.
Mhm.
Fundamentally, if you're chasing the wrong target, you fail. And so target discovery is where we're allocating our effort, using all the available tools, the massive expansion in human data, to create a platform that is about sophisticated, realistic, ecosystem level target identification, validating those targets in our assays, are truly representative assays, and then using AI tools to accelerate the drug discovery process. And so really, my money is on best target identification. We are stuck in the muck when it comes to target identification, chasing the same targets over and over again, or very similar targets. And that's where we need to unplug, reset, and consider everything we already know about the system of the tumor for best target identification.
You were at JP Morgan, you attended Bio in Boston.
Mhm.
When you look at the reality of capital allocation, both from the industry and venture capitalists, where do they put their capital these days?
Um, it's looking to me just from the perspective of bio, um, the a lot of the larger pharma companies are really interested in partnering with smaller startups. They see it as the way to best educate their internal teams. They see it as the way to become smarter. They see it as a way to to grow without excessive risk by engaging with startups that are taking a lot of risk. And I think that's a really smart allocation of their resources. So the big pharma companies each have their own program for doing that, which I think is really interesting. I also just see a lot of technology deployment. There's a lot of really interesting work going into simultaneous profiling for tumors, where one sample has single cell sequencing done and RNA sequencing done and every other kind of in proteomics, so that each patient has a complete profile. So I'm seeing some allocation in that direction as well, which I think is really powerful. That's the data we want to be using is multimodal
Mm.
patient data. So, I think there's a lot of wisdom around those plays and those decisions. And then I see a lot of the same old, same old. There's just, the sector in general is still looking at best checkpoint inhibitors, best GLP-1s, a lot of the same tried and true, which, probably has economic value to it, but is beyond my area of expertise.
But nobody seems to do what you try to achieve. So
No.
this is really the really high novelty in your approach.
Yes, we are alone out on this limb. And I think I'm glad to be there. I'm happy to lead the way. My dream is that in 5 years, everybody will be out on that limb with us. Uh, you know, in terms of what benefits patients the most, having the most tools in this direction. I can't do it all myself. That's a ridiculous notion. I want to create a community of systems level cancer biologists who don't just accept the complexity of the solid tumor, but thrive in it. We have to lean into it. We can't be afraid anymore. We can't work around it anymore. We simply have to dive in. And I guess I'm just here to say the water's fine. Come on in. It's not that scary. You can do it. And I think I really do feel like we would all agree that the tumor ecosystem is the final frontier. It is what's standing in our way, and we can solve this problem.
Karin, great conversation. Is there, before we wrap up, is there anything open that's, is there any question I should have asked?
I feel like you asked a lot of good questions. I think if people want to talk about it further, if they have questions, they can always reach out to me through my LinkedIn page or through our website at syzonc.com, SYZONC.com, and we're happy to continue the conversation. I think it's an exciting time to be a cancer biologist. We're at this incredible precipice, and we really, really have high hopes for the future.
Karin, thank you very much. You took away my last question and you answered it without me asking it.
Sorry.
What's the, no, no, that's fine. So what's the best way to reach you? LinkedIn, you're responsive on LinkedIn.
Yeah,
And
that's easiest. Yeah.
Karin, thank you very much for this great conversation. I learned a lot of new things about the last frontier in cancer product development. I hope that you succeed because from my own experience, I know that a lot of patients are waiting for a solution that really works and that makes all the other therapies work. And I hope
Mhm.
you find the investment and your team keeps pushing forward regardless of the resistance they receive from the environment.
Thanks very much. Appreciate it. And thanks so much for having me. This has been great.
Sure, thank you for